Hello everyone, and welcome to our webinar entitled Supporting Gut Barrier Function. Our guest speaker today is Dr. Thomas Guilliams. My name is Michael Chapman, and I'm a medical education specialist at Genova's Asheville branch, and I'm going to serve as the moderator for today's webinar. And we'd like to welcome Dr. Tom Guilliams. Dr. Guilliams earned his doctorate in molecular immunology from the Medical College of Wisconsin in Milwaukee. For the past 2 decades, he has spent his time investigating the mechanisms and actions of lifestyle and nutrient-based therapies, and is an expert in the therapeutic uses of dietary supplements. Thomas serves as an adjunct assistant professor at the University of Wisconsin School of Pharmacy and is the VP of Science for orthomolecular products. Since 2014, he has been writing a series of teaching manuals that outline and evaluate the evidence for the principles and protocols that are fundamental to the functional and integrative medical community. He is the founder and director of the Point Institute, an independent research and publishing organization that facilitates the distribution of his many publications. A frequent guest speaker, Dr. Guilliams provides training to a variety of healthcare disciplines in the use of lifestyle and natural medicines. He lives in the woods outside Stevens Point, Wisconsin, with his wife and children. One of the most common questions we get asked during the webinar is about availability of this presentation in the slide deck. These materials will be available on our website within a week of the webinar, and if you're interested in having these resources, please click the Clinicians tab on the home page, where you'll find access to our webinars under Medical Education, or you can also log to your LiveGDX account to find these webinars. If you do not have a LiveGDX account, please click on Getting Started link on our home page. And now I will turn it over to Dr. Guilliams.
I appreciate the opportunity to give this webinar today on a topic that's obviously very interesting and very important for the functional medicine community and obviously the patients that they serve. So as was mentioned, this is information that comes out of one of the recent books. This was published last year, Functional Strategies for the Management of Gastrointestinal Disorders. We're going to cover not all of this, obviously, but you can see on the right-hand side the 5 main areas that are covered within this book from a functional standpoint: digestion and absorption, the microbial ecosystem of the gut, neuroendocrine functions, elimination and detoxification, and of course the protective barrier or interface is what we're going to kind of cover today. Obviously, in the 45 minutes or so we have, I'm not going to cover every detail, but I want to give a highlight of kind of what we know about where we can influence the barrier function of the gut. We can test for that, and then the very limited information that we have about things that we know function in changing the barrier function of the gut in ways we can measure. So the idea of a barrier or interface is really, universal to all biology. Almost everything happens at an interface, whether it's the discrete different functions between the inside and outside of a tissue, a cell, an organelle, a gene. A gene even has a beginning and an end. We know there's interfaces between the promoter region, coding region, etc. So these interfaces are where we have all of the organization, all of the control of what happens in and outside of that system. So, and in order to have a good interface, we have to have a barrier. So if there is no barrier between one side and the other, the interface obviously is of no use. So if you have a exquisite gate in a wall that is supposed to control what happens on each side of the wall, obviously that doesn't work if the barrier is completely destroyed. So if you go back to looking at these interfaces or these places where we look at larger pieces of the way the body looks at surveilling risk or risk from the environment, you can see these are 3 areas that I spend quite a bit of time on: the HPA axis and the stress response, the immune system, and the GI system. And if you look at how they interface with one another, each one of these is a broad interface that allows us to deal with, in the case of the HPA axis, stress from outside the body, and getting information from the inside to kind of give us this idea of an overall stress response. The immune system is supposed to give us this interface between self and non-self. And finally, the GI tract is really this large interface with the outside world, as we're going to see, and it signals and coordinates the immune system and the brain and really all three of these are related together. And interestingly, if you look back at Hans Selye's work on the stress that he put in animals, he defined his stress response as an alteration in the HPA axis, including hypertrophy of the adrenal gland, atrophy of the thymus, so a change in the immune system function, and erosions in or ulcers in duodenum, so change in the GI system. So this was really the coordinated definition he had, and he obviously looked at a number of different things that said that this was quintessential to a dysfunction in the stress response or the surveillance systems of the body. So it's not coincidental that these three are tied together, and it's one of the reasons why I always see these three areas, the HPA axis, the immune system, and the GI system, as being highly coordinated in chronic disease management, and in many cases acute disease management as well. So as we think of an interface, especially when it comes to the gut, we want to think of a couple different things here. I wrote this I think in the beginning of the book, or in the beginning of the barrier chapter of the book, that talks about not imagining the barrier function as meaning just something that keeps things out, but a sophisticated system to communicate with and allow selective entry of certain contents from the gut lumen into the body, requiring a tight control, but a thin barrier of tissue and secretions intentionally designed for close proximity to the gut lumen. One of the things that we need to think about here is that if the gut wanted to be just a barrier, it would be a much better barrier. The reason that the barrier function of the gut is so tenuous sometimes is because it's designed to have close proximity to the gut lumen, because it needs signals, it needs nutrients, it needs access to that environment, which creates vulnerability to those same microbes, toxins, and immunologically reactive components. So I think that's really an important feature of this concept. We probably need to eventually use the word interface even more so than barrier. So obviously the gut is designed for that system, and one of the ways it does that is by greatly expanding its surface area. So we see not only the GI tract creating these large villi, but then we have these microvilli that greatly expand the surface area. If you just take the cells themselves, people say, it's about the size of a tennis court or a doubles tennis court. Obviously, that's a huge area for which the immune system has to spread out and protect, sort of like a castle wall. There has to be sort of a way to protect the whole thing all the time. When we have a vulnerability, in the case of, an extreme case like celiac disease, where we have hyperinflammation, we see villus atrophy. We see an atrophy of the surface area, and obviously that has negative connotations and negative consequences, but that actually is an adaptive response to the protective mechanism of the barrier function. If you have a tennis court and you can shrink that down, into half that size, you have half the barrier to protect against. And so again, this creates a benefit for some of the interface functions, but a greater vulnerability as well. So if we think of the functional components of the gut barrier, we have human GI cells. We're going to talk about those. Typically, we think of those as the enterocytes and the colonocytes. We think of that as this, quote, single layer of cells that protects us, the gut lumen, from the inside of the body. We have obviously the whole human immune cells that line inside or penetrate the interface itself. So we have those cells that we want to think of as part of the gut barrier. We have neuroendocrine cells and neurons with synapses nearby, so they actually help communicate the barrier function to the brain, and the brain to the barrier function. So we have some interface with neuroendocrine cells. We have secretions from many of those cells. So we have secretions like mucus, secretory IgA, antimicrobial peptides, enzymes, acid, neurotransmitters. These are all secretions that become part of the gut barrier itself. And finally, we have non-human microbes in the lumen and mucus lining. And so we think of these as obviously commensal or pathobiont or potentially pathogenic bacteria, viruses, both free viruses and bacteriophages that are in bacteria, fungi, non-human eukaryotic organisms. Maybe some of those will be determined to be commensals. So there, as we will see, we don't have a lot of time to get into the microbiota component today, but there is quite a bit of information on how they influence both beneficially and potentially negatively the gut barrier function. So as you look at this image, and this actually is a graphic from the book itself to show sort of the basic features of, on the left-hand side, the small intestinal gut barrier, and on the right-hand side, the colon gut barrier. You can see some differences. The small intestines are notably have a greater surface area with much larger villi, designed mostly to bring in more nutrients. So we have both crypt and villi in the small intestine, where, as you can see in the colon, we mostly have crypts, but not really many villi. The other major difference between the two is on the left-hand side, you'll see a single layer of mucus, which is kind of fragmented, which is sort of the gray, sort of looks like bricks there, fragmented mucus, because we need to have more access to the cells in the small intestine, where in the large intestine we have two layers of mucus, a inner layer of mucus, which is very viscous and very difficult for bacteria to penetrate, an outer mucus layer, which is less viscous, which is a little more easy to penetrate for bacteria, and then the gut lumen, where you have most of the bacteria in the colon. And there's a very definite difference between these two. The small intestine is designed as an interface that is very absorptive and needs a lot of interaction with the immune system and with nutrient absorption, whereas the colon, there is some interaction obviously with the immune system, much less absorption, and it really acts primarily, or in some cases primarily, as a fermentation for the ecosystem, the microbiome that's there. So there's quite a bit of a different function, and therefore you're not surprised to see a different sort of a feature of the barrier itself. So let's just talk about some of the cells that make up the barrier. Stem cells are very important. They sit at the nexus of the crypt, and they're essentially there to produce almost every other cell type here. So they are dividing, and as the cells are moving up the villi, they are not only dividing, they're differentiating, and they're differentiating into goblet cells that produce mucus, or some of the enteroendocrine cells that produce some of the hormones, or the absorptive cells in the case of the enterocytes, the ability to produce enzymes and or transporters. So you have these going up the villi, and every 24 to 36 hours you're seeing these cells turn over. They move up out of the crypt into the villi, and they're being shed every 36 to 48 hours. And so this is why the enterocytes and the gut need so much energy, and they need so many building blocks, is because it's a constant turnover. And as you can imagine, every time a cell is formed, there needs to be a new set of tight junctions formed. And so that's why tight junction proteins become so important. As you're producing these cells, you have to obviously produce the cells and their connectivity to the other cells. Now, this is just showing this in 2 dimensions. Obviously, it's in 3 dimensions, so it's actually going really always, towards you and away from you at the same time. The other cell type that we have here is the Paneth cell, which are shown on the right-hand side in green and in the left-hand side in yellow. And these actually go into the crypt, and they produce quite a few of the antimicrobial components that are there in the crypt, because the crypt is a very protected space. As it turns out, the Paneth cells, I'll mention here in a minute, are also important for maintaining stem cell function itself. So we have really, from these stem cells, we make all of these different sort of specialized cells. So we know quite a bit about the absorptive epithelial cells. I'm not going to spend a lot of time talking about the details. I'm going to mention a few things about them, but they're located obviously primarily in the duodenum and the ileum and jejunum, and we have a lot of these, the ability to move a lot of nutrients through the transcellular route in a controlled fashion. To protect these cells, obviously, the tight junctions become important. So I'm going to spend a little bit of time explaining the tight junctions for those of you who aren't as familiar with the proteins that are involved. On the left-hand side, you can see this picture that gives you a better understanding of what the tight junctions are there for. They don't actually close off the cell-to-cell junction completely. They actually end up creating these very small pores which allow water and a few small electrolytes to get through. And they're not just a single connection. You can see it's almost like a series of Ziploc. If you can imagine a series of Ziploc closures with tiny little holes to allow water and electrolytes through. That's what it's supposed to create. And it's usually up on the surface, the luminal surface. You'll see most of the tight junctions. On the right-hand side, you can see that it's basically created by these proteins that act as little, almost like Velcro. On one side, you have the claudins and occludins. On the other side, you have other claudins and occludins and other proteins that are connected. And they essentially can overlap one another and create this dynamic junction. And they're connected on the inside to what we call ZO1. You can see zonula occludens. These are types of proteins that connect these claudin and occludens together, and they are in turn connected to myosin and actin fibers. And so what happens is they are all sitting, as if you can imagine, sitting together like a Velcro connected to, let's say, wires or rubber bands, that if those are pulled back, then you can get an opening of the tight junction. So instead of having small aqueous pores, you end up having much larger pores. And one of the ways that happens is a kinase reaction or phosphorylase reaction that's driven by something called myosin light chain kinase, and MLCK it's referred to here. And when that activity phosphorylates the myosin chain, it contracts and pulls back and opens up these junctions. And one of the main ways that happens is inflammation. There's several inflammatory cascades which will cause these to open up. And we're going to talk a little bit later about how we see that with zonulin, and maybe ask the question about why do these cells have the ability to do this? If this was never intended to be opened up, why is there a mechanism by which kinase can, in this case, inflammation, actually open this up, what seems like intentionally? And most people believe it's to give the immune system more direct access to the lumen. Unfortunately, the reverse also is true. The lumen has more access to the immune system. So digestion and absorption occurs mostly in the upper GI, and we have very specific places where absorption of certain nutrients occurs. One of the things that's very important is that to understand that it's a little bit difficult to see perhaps in the slide, but disaccharidases, the enzymes that break down maltose into its glucose components or lactose into its monosaccharides, are located on the brush border. They're actually located not in the lumen, but they're actually located on the cell surface, and that occurs when you have healthy enterocytes. However, if you have an inflamed individual and they have villus atrophy and they lose the number of cells that they would have otherwise be there, you actually reduce the capacity then to digest disaccharides. You don't lose it completely, but you reduce that capacity. So you oftentimes will see people who become lactose intolerant or intolerant to other disaccharides as a secondary effect to other inflammatory GI conditions, because you've lost some of the benefit of the capacity of these disaccharadases. You also, as you can see in the lower picture there, you have a number of enzymes that take small polypeptides and break them down into amino acids. And you have a number of transporters as well. So you'll also perhaps have the inability to break down certain small peptides, and also then drive potentially other immunological reactions that will be secondary to inflammatory conditions. So it's really important to understand that the enterocytes are not just there to absorb nutrients sort of passively, that the transporters and the enzymes that are involved are highly important for, especially in this case, macronutrient absorption. Lipids are a little different, so they don't require enzymes per se from from enterocytes, but certainly disaccharides and small polypeptides. And so this becomes a very important component. So again, let's run through a few of these others. Paneth cells, you know, again, found only in the small intestines, found primarily in the ileum. They migrate into the crypt after differentiation from stem cells, so they move down rather than up. And they are the primary secretors of antimicrobial peptides in the gut lumen. And so there's a different sort of cells. There are actually a few other cells. Enterocytes themselves actually can produce some antimicrobial peptides, but in this case, the Paneth cells are there. They're very long-lived, so they don't turn over nearly as fast as those that are migrating upward. And then they specifically help regulate the stem cell activity. So they're highly regulatory, and that's why it's important that they function in the crypt and aren't, you know, so vulnerable to other stressors in the GI lumen. The immune system is tightly bound to the GI cells. On the left-hand side, you can see generally where you have these Peyer's patches, and I've shown also that you have epithelial cells, these intraepithelial lymphocytes. On both sides, they're in blue, and essentially they tuck themselves in about every 10 to 15 cells. They are there, and so you see them interspersed all throughout. And essentially, these are very unique kinds of T cells, and they slightly different because they're more regulatory type T cells rather than effector type T cells, and they're there to continually sense what's going on. Dendritic cells are able to put their arm all the way through into the gut lumen and begin sampling what's going on. Dendritic cells are really the organizers of the immune system within the gut. And then on the left-hand side of the right picture, where you see these M cells are called microfold cells. These are special kinds of enterocytes designed specifically to allow antigens to come across and be available in a controlled environment to both dendritic cells and mostly T cells. So we have T and B cells and dendritic cells that are all sitting there sampling, constantly sampling what is available to it in the gut lumen. This is mostly occurring in the small intestines, where you have much more accessibility to the M cells. You have very little of this going on in the colon. So again, the colon is designed to be, let's say, more of a barrier than the interface functions of the small intestines. So there's 2 layers of mucus, as we mentioned. There's an increased number of goblet cells because they're the ones producing mucin. There's, like I said, less interface, more barrier, much fewer or a much lower concentration of immune cells. We have what we call colon patches rather than Peyer's patches. They're much fewer and much less concentrated in their ability to function, but obviously they're there because you do have breaches in the colon as well. There's fewer endocrine cells, so there's less cells that are signaling because there's just less of that going on here, because you have really more of a large capacity for fermentation. So you have really a controlled fermenting environment rather than a controlled interface environment. And one of the ways that that's controlled, and I talk quite a bit about this in the book in our microbiota or microbiome chapter, is the idea that especially in the colon you have these different layers. The lumen, which has the large amount of bacteria. And you have some that penetrate into the outer mucus layer, and very few. These are usually flagellated bacteria or those that have very special characteristics that are able to get all the way down into the inner mucus layer of the colon. And this is controlled by a number of different factors. I speak about in the book quite a bit about the different mucins that make up both the firm and the loose mucus in the gut. There are certain bacteria that actually are intended or embed themselves into the mucus. Akkermansia mucinophilia is one classic example where we have a good commensal organism that embeds itself in the mucus. There's different shear gradients, so obviously as the contents of the gut lumen are moving, you have different shear forces which affect which bacteria can survive. Different oxygen gradients also affect the ability for different bacteria to survive in different areas of the gut, the availability of host defense molecules, the concentration of those, the ability for the immune system to produce specific secretory IgA, et cetera, et cetera. And as we mentioned, the change in nutrient within the mucus with Akkermansia, and then the crypt niche, which is extremely, extremely special and needs to be protected. And so, like it's showing here, only a few bacteria ever make it down into that. And there's a lot of data on, looking at, you have to obviously do this by biopsy to look at different areas, not only longitudinally along the gut, but then, sequentially or cross-sectionally across the gut. You have different microbiota in all these different areas, and all of those are influencing the gut barrier itself. So breaking the gut barrier, and so the term leaky gut, as many of you know, is becoming, I guess, in some ways more popular. I showed on the right-hand side, I did this briefly, looked at leaky gut on PubMed over the last, since 1984. And you can see, while it has grown quite a bit in the last 4 or 5 years, we still don't have that many hits per year. I mean, the highest we have is 26 hits in the last 3 years, and it really in 2010, 2011, it just exceeded 10. So this term is still not used that often. And I pulled this from WebMD. I thought it was kind of interesting. It says here, from an MD standpoint, it's a very gray area. Says GI gastroenterologist Donald Kirby. Physicians don't know enough about the gut, which is our biggest immune system organ. Leaky gut syndrome isn't a diagnosis taught in medical school. Instead, leaky gut really means you've got a diagnosis that still needs to be made. You hope that your doctor is good enough Sherlock Holmes, but sometimes it's very hard to make a diagnosis. We don't know a lot, but we know that it exists, says Linda Lee from Johns Hopkins. In the absence of evidence, we don't know what it means or what therapies can directly address it. So as you know, if you get a hold of the book, you realize I do like the term intestinal permeability a little more because, and maybe other specific terms, but certainly leaky gut is a term that has, unlike other terms, maybe is growing a little bit, and people are a little more comfortable with it as we know more about it. Here's a couple papers just came out in the last couple of years to kind of give you an idea. Leaky gut as a danger signal for autoimmune disease. And on the right-hand side, elevated fecal calprotectin in patients with Alzheimer's dementia indicates leaky gut. So we're starting to see, other people use this in different terms. So it's a term that still has some controversy behind it. Anyway, one of the reasons is because some people view leaky gut as only this extreme view. And essentially what we mean by that extreme view is a view where basically, whole organisms are getting in past the gut because there's the complete breach of the barrier. And we measure this by, sepsis or systemic infections or, lipopolysaccharides or some, endotoxemia kind of thing, rather than perhaps a more subtle view that we're seeing where we're just seeing small immunogenic agents getting through or alterations in the immune function caused by changes in tight junction function. So we know that there's various ranges of what leaky gut can mean. If you look at a more common scenario here where I'm showing some sort of inflammation, you can see at the top, this MLC is the myosin light chain kinase that is phosphorylating the myosin light chain, which is then pulling back our occludins and claudins and pulling back and opening up. You can open up this the tight junction, creating this intestinal permeability. And there's many different things that can do that. And of course, gliadin or gluten is sort of become one of the quintessential ways to look at this through zonulin, and we'll look at that briefly here. So as many of you know, Alessio Fasano did a number of, a whole range of research in this area, looking specifically for cholera, a cholera toxin molecule actually, and came across a molecule that he ended up coining as zonulin, We also know that it actually is part of another molecule, but zonulin became the name that he gave to it, and he expanded this story looking at the role of zonulin and gluten or gliadin as a trigger for the whole leaky gut or intestinal permeability component. For those who aren't as familiar with the story, essentially, and we don't have time to get into this in detail today, but essentially what happens, and basically from this picture from left to right, up at the top it's showing gluten or gliadin protein or peptide binding to a protein on the cell surface, the CXCR3 receptor, and that triggers in those cells, or in some of those cells, depending on their genetics, a high amount of zonulin. Zonulin is, again, this protein, still a question about why we produce this protein. It's called prehaptoglobulin, so maybe it has another function in the body. But as zonulin is produced and secreted, it can bind to the cell surface of enterocytes, and as it binds to the cell surface, it then triggers myosin light chain kinase, which then pulls back the tight junctions, or pulls back zonulin includin 1, which then pulls on occludin and claudins, and opens up this junction. And you can see here, obviously, this allows peptides. In this case, it's showing gluten or gliadin peptides coming in and binding to T cells and creating in certain individuals the whole celiac phenomena, and perhaps in other people non-celiac gluten sensitivity. And all of that going on is, the question is, why does the body have a system, why are these cells have a system that allows the tight junction to open up? And so many people are speculating that in certain instances, the body actually creates a triggering signal, perhaps from certain peptides or bacteria, that triggers the opening of the tight junction, intending for the immune system to have greater access to the gut lumen in order to protect itself. However, perhaps in an overabundance of certain peptides like gluten, that this backfires and we end up having too much activity, too much immune interaction with the gut lumen, creating the autoimmune cascade that we see with celiac, but also other related autoimmune diseases that may be triggered. So how do we measure this? How do we measure the permeability? So there's a number of different ways to do this. The gold standard is actually to do an ex vivo biopsy of the gut cells, and you place them in what's called an Ussing chamber. And this picture on the left-hand side essentially shows, it only shows one single cell, but it would imagine there's a series of cells all oriented with the luminal side on one side. So you have all of them oriented the same way. And now you can measure their ability to allow, in this case, it's showing chloride ions, but you can basically measure the transepithelial electrical resistance. So you can measure how easy can we get this barrier to allow electrical resistance. And so if there's no resistance, obviously it means that we have a breach in the barrier. We have a very poor barrier function. And so this became sort of the gold standard in not only can you do this with biopsies, you can actually just grow cells along an Ussing chamber, and then you can test, botanicals, you can test other noxious agents and see how they affect the gut barrier. So a lot of the data that we know about certain agents that either improve or decrease gut barrier function occurs in ex vivo Ussing chamber type reactions. So it's something that you want to keep in mind when you look at the research. We have less in vivo ways to do this. The one in vivo measure that is, I think, generally thought of as the best currently is the size exclusion test, which is typically thought of as the lactulose mannitol test. It's probably the most common. And essentially this allows you to put different size carbohydrates in the system, lactulose and mannitol, whereas mannitol should be able to get through the cell surface through the normal transepithelial pathway, gives you sort of a big picture of what is the denominator, what is the gut area, and lactulose should not be able to get easily across unless it's going through between the cells, So if you have lactulose in your urine or a high level of lactulose, it means you're getting more and more of that through between the cells. So this lactulose to mannitol test becomes a general ratio that allows you to give an idea of, what is the ratio between sort of the overall surface of the gut and the ability for lactulose to get through in a paracellular manner. There's a few others that are used as well, but the lactulose mannitol is probably the most common. So other tests that are available or thought to have an effect on gut permeability: urine or serum levels for microbial metabolites, endotoxin, D-lactate are often can be used. Increased levels of bacteria in the dense mucus. That you actually can biopsy the dense mucus and show that the more bacteria in the dense mucus of the colon, the more likely you are to have a intestinal permeability. Reduced plasma citrulline is a biomarker of glutamine. Fecal calprotectin, as we've seen, has also been shown to be connected with that. It's mostly obviously an inflammatory marker, but very linked to gut permeability. And you can also measure zonulin, Gliadin, occludin, and look at tight junction proteins. These are often done ex vivo, but you can also look at these, and there's some data suggesting that. And then serum and maybe fecal zonulin is also being used quite readily now as a marker of intestinal permeability, or I should probably say a marker of gut barrier function, which may or may not result always in intestinal permeability per se. So we know that there are many conditions. Any condition where you're putting stress on the gut, in an inflammatory stress, we know less about, for instance, things like IBS. There are some studies that show IBS with diarrhea is statistically connected with changes in gut barrier function, although in other cases it's not, probably because IBS definition is not always as clear in different cohorts. And then there's some question about SIBO, whether SIBO as a diagnosis is connected with changes in gut barrier function. What we do know is there's a growing amount of research suggesting that obese subjects have more and more intestinal permeability. There's a number of different studies now looking at this, in this case, serum zonulin levels go up in most cases with obesity and insulin resistance. So something that we're obviously is a growing problem and should be looked at in those subjects. One of the issues we have in looking at the data on preventing and treating is that intestinal permeability is often not measured. Like I said, there's some of these studies that look at Ussing chambers, but very few human clinical trials actually look at this. So you start looking in the literature and you say, well, are there data on dietary interventions and changing gut permeability levels? Well, there actually are very few. I actually found this one that showed when they actually gave, now you may not like this, but they gave pasta, a wheat product, but they gave them a 5-week crossover study looking at healthy young individuals, giving them 800 grams of pasta with and without inulin, which is a prebiotic that stimulates good bacteria typically. And you can see they saw a statistical change in just 5 weeks, a statistical reduction in lactulose mannitol, a reduction in serum zonulin, and a slight increase in GLP-1, which is an endocrine component in the gut. So again, very few. I mean, I wish I had 100 papers to show you, but there are very few like this kind of study, and this was just done just a few years ago. If you look in the data and say, how about micronutrients, vitamins, minerals, all the things that we think are good for the gut, we have very, very limited data, especially in what I call healthy or relatively healthy Westerners. Most of these are done in either very sick children or in people that exercise intensively and see a drop in gut permeability or increase, I should say, in gut permeability and a decrease in immune function. So one of those that we often think about quite often is glutamine. For as many years as I've heard about glutamine, you'd think there'd be hundreds and hundreds of studies on the role of glutamine in barrier function. As it turns out, very few. Most of these are on burn or severe injury patients. It's frequently used with, I think probably many people that are listening to this would say, they've gotten great results using glutamine, but the research on it is very, very limited. when you look at animal studies and cell culture studies, there's a whole list of things. I'm not going to go down this list. You have the slides, but there are numerous explanations for why we think glutamine would affect intestinal barrier function. It improves almost everything we want to look at. This is a great recent review published this year on the role of glutamine in intestines and its implications in intestinal diseases. I would recommend that you get it, but you can see, the standard sort of things that we would think about, all benefiting and what we would assume maintaining intestinal tissue integrity. However, like I said, we have very few studies in humans. This study that I show you here shows that they took these Brazilian shantytown children that had high levels of intestinal barrier, or I should say low levels of intestinal barrier function, and they were able to improve thriving. They were able to improve their thriving, but they actually didn't really see much of a change, and they didn't really even look at the level of intestinal permeability. You can see below here a meta-analysis on critically ill patients looking at glutamine-enriched enteral feeding. So these are studies that are not, the typical ones that you're seeing. Here's a study where they actually looked at patients with Crohn's disease, and you can see they used both glutamine and saw an improvement in intestinal permeability, but look at the dose, 40 grams of glutamine. They gave them half a gram for every kilogram of ideal body weight. So this is for an 80 kilogram person, that would be 40 grams of glutamine. So this is, again, an extreme sort of situation. A few others that we have in the literature are during immune function, during heavy load training, given glutamine 10 grams a day. You can see that they were able to attenuate the immunosuppression, but they didn't actually measure gut permeability. So we can assume there was some benefit, but we don't know. So I tend to tell people that, the dose recommendation for a lot of clinicians is somewhere in the 4 to 8 grams a day range, which typically requires this to be taken not in capsules, but in powders. And again, large anecdotal data set out there, but very little clinical trials for me to tell you exactly if you see this in the lactulose mannitol test and you give this amount of glutamine, you're going to see this kind of change in this amount of time. We need more. And actually we need more people publishing their data so that we can get access to this information. We know that there's a lot of phytonutrients out there that I can't go through all of them, and I'm not going to try, but there's a number of them. I typically tell people phytonutrients should be thought of as a way to diversify the diet, not that you're trying to get a specific dose. It's really better to think of following a long-term low-dose supplementation, i.e. good diversity in the fruits and vegetables that you eat, and or the supplements that you can supplement with a diversity of phytonutrients. Berberine is one that's kind of come on the scene, which is interesting because there's been some study in animals suggesting that it can improve intestinal barrier function, and so we've seen some of this data improving tight junctions, improving in animal models, both type 2 diabetic animal models, so we see that effect when it comes to humans as well. Here's a study looking at IBS in humans, looking at changes in diarrhea given berberine hydrochloride. You can see a benefit on the left-hand side for the change in urgent need for defecation. They gave this for 8 weeks. You can see it continued down for 8 weeks, and after they stopped, it went back up. So whether or not this is related at all to gut barrier function, we don't know because they didn't measure it. And gut permeability is not often measured in some of these studies. Probiotics are often thought to be beneficial. Again, good commensal organisms are always acting to benefit the gut. There's a number of different studies that have looked at in vitro. These are in vitro studies, mostly looking at Ussing chamber type experiments. And you can see, bifidobacteria, Lactobacillus plantarum, VSL3, which is a combination of 8 different bacteria, Bifidobacteria infantis, and L. acidophilus, and at the bottom you can see Saccharomyces boulardii. I believe that if you look beyond these, you'll see that most lactobacilli and bifidobacteria and probably Saccharomyces, anything that improves sort of the status of the gut is likely to have some sort of triggering effect on improving the intestinal barrier function. There might be some exceptions to that, but I think that's going to be mostly true. A good gut microbiota is going to improve gut barrier function. We see a few different studies. This was actually a meta-analysis looking at a number of Chinese studies on patients going in with colorectal cancer for colorectal cancer operations, and they showed that they were able to improve the lactulose to mannitol test with almost every probiotic strain and dose they tried. So again, these are somewhat low quality studies, but kind of gives you the idea that when you start looking at this, there seems to be some improvement. Here was just one of the studies looking at 160 subjects given probiotics, and this was actually a combination of Plantarum, Acidophilus, and B. longum. And you can see that post-operative serum zonulin, if you look up in the upper left-hand corner, you can see bacterial translocation went down. At the lower left-hand side, you can see the transepithelial resistance. So this is a Ussing chamber study or test, and you can see that went up, so it improved. And up in the right-hand corner, you can see lactulose to mannitol test went down, and the transmucosal permeation, which means bacteria within the mucus, went down. So this was, again, all the things that they measured, which wasn't systematic, but they all improved with giving this blend of a probiotic. Here's another study looking at probiotic supplements. This contained 6 different probiotics: bifidobacteria, lactobacillus, etc. And they looked at fecal zonulin levels in these individuals. And you can see fecal zonulin levels did go down. And again, fecal zonulin maybe is not as consistent as serum zonulin as far as predicting function of the gut, but nonetheless, another study looking at this. Saccharomyces boulardii, in this set of Crohn's patients, you can see a very modest dose of Saccharomyces boulardii, 400 million CFU, not even in the billions, given every 8 hours though. So this was given quite frequently, and you can see that given Saccharomyces boulardii, these Crohn's disease patients that were seeing their lactulose to mannitol ratio go up went down dramatically after being given the Saccharomyces boulardii. So again, these studies are very limited, but they do suggest the things that we've been talking about. How about colostrum? Here's just another couple things. Colostrum has been shown to look at this. These are in athletes again, and if I can just give you one from a lifestyle standpoint, if you have someone that has gut issues, especially if you think there's barrier issues and immune-related issues, one of the ways you can exacerbate that negatively is to have them become, these athletes that strain and stress themselves. So these are, again, a classic way to improve or to induce, I should say, intestinal permeability and induce immunosuppression is, run a marathon. So they take a lot of these individuals and they look at ways they can improve that. In this case, colostrum was able to improve the lactulose to mannitol ratio and reduce fecal zonulin levels when given 500 milligrams with 500 milligrams of bovine colostrum per day. So I know that I've covered only a little bit of a lot of things, but my job was to sort of give this overview of barrier function and what we can do about it. So some of the things you want to think about, obviously avoid foods that are going to create intestinal permeability. NSAIDs are a real no-no. They're going to be a problem. So if you can all ask them to reduce or eliminate their use of NSAIDs, that's going to help you dramatically. Reducing stress, we didn't have time to talk about it, but stress obviously has a direct effect on the tight junctions. Avoiding strenuous physical activity, avoiding processed foods with artificial colors and flavors. Often these inflame or cause the same problems. And eat abundant amounts of fresh fruits and vegetables. Maximize phytonutrient diversity. And then the list of things that I have here are a little bit broader than what I mentioned, but you can see glutamine, vitamin D, omega-3 fatty acids, pro and prebiotics. Zinc can also be shown. We know that zinc deficiency can increase intestinal permeability. Only use iron if you've shown that they do have iron deficiency. Don't just use that prophylactically. Flavonoids, colostrum, and then consider potentially berberine, especially if you have a subject who is insulin resistant and has type 2 diabetes, where that would be beneficial for other reasons. So I went fairly quickly, and hopefully I've covered enough that we can get some questions or at least follow up later on. And then I'll hand it over to you now, Michael.
Great. Thank you, Dr. Williams. Just a terrific presentation. We've received a lot of feedback already, and I just want to remind everyone that the PowerPoint will be available on our website in PDF format next week. We did receive quite a few clinical questions or general questions, so let me just start with, there was a question around research indicating that the potential of overdosing or using too much supplementation with probiotics might lead to increased immune load. I didn't wonder if you had any comment or if you've run across that.
So there are a lot of clinicians that I think have been told or just assume that the use of probiotics when you have somebody that let's say has high lactulose to mannitol or suspicious of leaky gut, that we should avoid that because all of a sudden we're going to create endotoxemia or some other kind of thing. I think, first of all, there's no evidence of that. And two, we would never imagine that making the gut aseptic is the solution for a good barrier. So as it turns out from the data that we've seen, and I think just logically what we know about good commensal organisms, that the use of probiotics is not contraindicated when somebody has intestinal permeability or leaky gut, and in fact may actually be beneficial. So if you believe that you're getting endotoxemia because you've got this major leaky gut, we're not going to give antibiotics to wipe out the gut. So adding good bacteria is certainly not going to be a problem. It's never been shown to be a problem, and the limited data we have suggests that it's actually beneficial.
Great, thank you. Another question around whether you've seen any research suggesting an impact of Lyme's disease or other tick-borne diseases on gut barrier function.
I have never specifically looked at that. I would not at all be surprised if there are some data suggesting that Lyme or tick-borne diseases, or really any immune, anything that would affect the immune system that dramatically would have gut barrier changes. But I have not specifically looked at that or seen that reported. But it would not at all surprise me.
Another question about how long, kind of a timeline, do you think that it might take to really restore, repair an inflamed GI tract, and even bring about better nutritional status and digestion absorption status?
So, I mean, obviously that's very patient specific. If we step back a ways, now I don't want to make it seem like the gut barrier becomes everything. If we back up and look at obviously the whole idea of what's going on in the gut, and the traditional sort of 4R approach and all these sort of things, it really depends. If the first R is to remove, and you've got a patient that is basically sabotaging themselves with their own diet routinely, and they're exposed to other things in their diet and alcohol and other things, and you're constantly seeing assault on the gut, how long is it going to take them to stop doing that? So in one subject, you may be able to get them to go cold turkey away from things that are harming them. Others, it may take a long time. So I think that's a huge part that I think people don't realize. Glutamine and but phytochemicals and probiotics in the midst of a diet that is constantly assaulting the gut is going to take forever. In fact, it may never benefit you. So I think it really depends. I think if you have a compliant and a person that's willing to change their diet very quickly, I think you can start seeing, things. Some people, obviously, within 3-4 days, you'll start seeing some dramatic changes in some symptomology. If you've got major breaches in the gut and history of antibiotics and things like that, it may take a while to rebuild. But I would think you're going to see a consistent increase month by month by month. And in some cases, I think you should, if you're not seeing anything by 6 months, you probably are missing something. You might be missing a parasite that you didn't recognize or some other thing going on. And then I think the issue is not the gut repair process, but really a diagnostic issue. What are you missing? And so I think it's going to be different for each individual, but it's not going to be instantaneous. And these takes months. In some cases, clinicians will tell you years to really get to the point where the gut is not vulnerable, easily vulnerable to, just one, off the wagon kind of thing.
Great. I found it interesting. You mentioned that the large intestine really kind of acts as a fermenting vessel. And so what are some of the changes that you might see or expect to see in the development of small intestinal bacterial overgrowth, and kind of your thoughts around how the etiology, how that might develop.
So if you and this is a kind of a tricky subject, but if you look at small intestinal bacterial overgrowth, and what we do to define it. Now, again, the gold standard for that is actually, aspiration of the duodenum or the ileum and actually counting bacteria and getting a number and saying that we actually have an overgrowth in the small intestine. The other way to look at that is a breath test. And a breath test is, and we don't have time to get into that, a breath test positive can be SIBO, can be overgrowth of bacteria, but it can be anything that creates that fermentation early, which can be bowel transit time, which can be even certain bacteria in the small bowel, whether they're overgrown overall or not. So it gives us some idea that something is going on, and we have symptomology based on, sort of this fermentation process. So the small intestine should not be a fermenting vessel, typically. And typically the lactobacilli and the lactobacilli that reside there and other bacteria that reside there are there typically doing other things. And one of the things that they're doing is they're having much more interaction with the immune system. And so that's why even small amounts of bacteria, lactobacilli, we say, well, it seems like a drop in the bucket compared to what's in the colon. But if they're in the small intestine, we don't need high doses. We don't need high amounts that are there because they're doing other sorts of things. So we certainly, if bowel transit time is slow, if acid is low, if other things are going on, you can get this migration of bacteria from the colon into the small intestine, which creates problems, some of which we can measure when we do either the glucose or lactulose test for the breath, but may or may not be SIBO in many of those cases.
Great. Yeah, and I know that was a very large topic, probably its own webinar. We had a lot of questions around a lot of questions about the book. So I just want to give you an opportunity to inform the audience about where they can get the book and speak to that.
So the book is published through the Point Institute, and you can purchase the book through what's called the Lifestyle Matrix Resource Center, LMRC we call it, the Lifestyle Matrix Resource Center. And so it's if you look it up online, Lifestyle Matrix, you can find it there, and you can purchase it directly. I don't know if Genova actually is, if you can create a link for that and let people have a link directly to that. The book is over 250 pages, or actually 250 pages, basically divided into those 5 different areas. So fundamentally teaching each of those principles. Like you can see some of the figures I showed, is basically the teaching manual for GI function. And then there's a series of monographs at the end that go through things like SIBO, IBS, IBD, H. pylori, et cetera, and going through sort of summarizing the evidence for the traditional and the functional approach to treating those. So the goal of the book is really to bring together the evidence for the common functional medicine approach to these. And in some cases, we show the evidence. In some cases, we suggest, like we did today, that there's good, fundamental mechanisms, but we don't have what we'd like to have as far as, a deep clinical trial evidence for some of the things that we often use.
Great. And just thank you so much again. In the interest of time, we're going to end our question and answer period there. For any additional educational materials, we'd like to encourage you to visit our website, www.gdx.net. On this site, you'll find sample reports, kit instructions, and other information for all of our profiles. We'd also like to remind you of our upcoming microbiome symposium on October 28th in Washington, D.C. It'll be a full-day event with Dr. Todd Lepine, and you can register at attend.gdx.net. Finally, we'd like to encourage you to look for upcoming webinars on www.gdx.net. Next month, we have our very own Dr. Doreen Saltiel presenting on advancing patient management, the role of genetics in cardiovascular disease. And just thank you so much again, Dr. Guilliams, for a superb presentation.
It was my pleasure to be with you.